Hello everyone, my name is Fahim Rida and now I am going to talk about RNA vaccines for infectious diseases. So really, the first question we are trying to answer here is why we need new type of vaccine. So what is wrong with the platforms that we already have? So really, I sum this up in kind of 6 main categories. So the first is really the lack of infectious treatment for many devastating infections, the emergence of multi-drug resistant bacteria. We are always looking to improve the safety profile of licensed vaccines. Obviously, disease outbreaks is quite a hot topic right now. We never know when a new virus will evolve and we will need to make a vaccine to it. There is also the changing age structure of the population, and the last is reduced cost and the time required to produce vaccines. So RNA vaccine is really just defined as the use of RNA to express an antigen and then induce an immune response. The problem with DNA and RNA vaccines are that it requires the delivery vehicle to get the nucleic acid either into the cytoplasm in the case of RNA or the nucleus in the case of DNA, but also to protect it from degradation while it's making that journey. So there is a number of advantages of nucleic acid vaccines that make them a really cutting edge technology. So it's really a speed of development. They are also much easier to make than proteins and live attenuated organisms and whole pathogens. It's generally a cheaper per dose and you can encode any antigen. They are generally safer and less inflammatory. They can be quite thermostable. They enable both translational modification, so the specific glycosylation patterns that are found in mammalian cells because they are made in the human cells, as well as the ability to express membrane-bound proteins. So now I'm going to delve deeper into self-amplifying RNA vaccine. So this is a specific type of RNA that we use, and I think it's really just useful to compare it to both plasmid DNA as well as messenger RNA. So plasmid DNA is a double-stranded circular nucleic acid. For the contracts we use, it's usually around 7,000 per. For the messenger RNA, it's a single-stranded linear construct, and for our typical construct is around 2,000 nucleotides, whereas the self-amplifying RNA is much larger, so similar in structure, single-stranded and linear to the messenger RNA, but usually around 9,500 nucleotides. So it's much bigger molecule. However, if we compare it to plasma DNA, it doesn't require penetration into the nucleus, so it only has to cross one membrane, which make it a little bit easier to deliver, and there is also no risk of integration into the host genome. So there is a typical structure of the self-amplifying RNA. So we have the 5' cap and the poly A tail. This really help protect it from degradation. Also an untranslated region at both the 5' and 3' end. These are integral for the replication of the RNA. We then have these 4 non-structural protein, and these are what make up the replicate, so this is what gives it its self-amplifying properties. So it's derived from an alpha virus that uses it to replicate the virus's own RNA, and we have kind of just hijacked the system and put it into messenger RNA for our own use. So then downstream of the subgenomic promoter we have here, and we have antigen code. So this schematic then show just the difference between self-amplifying RNA and messenger RNA when it actually gets into the cell. So if you look at the messenger RNA, once one copy is able to cross the membrane, then you have it in the cytoplasm and that gets translated into a protein. However, when self-amplifying RNA is able to cross that membrane and it gets into the cytoplasm, you get many copies of the original RNA and thus much more higher translation of protein. So, how does an mRNA vaccine work? To develop a coronavirus vaccine, researchers selected an mRNA that encodes the coronavirus spike proteins. These proteins help the virus enter a human cell. First, the mRNA is prepared by in vitro transcription or chemical synthesis. Next, the mRNA is encapsulated with lipid nanoparticles. This layer protects the mRNA and raises a stronger immune response. The final vaccine formulation is injected into the skin or muscle. Once the mRNA is entered into the cell, it uses the cell's translation machinery to produce the viral spike proteins. The spike proteins trigger the body's immune response to produce antibody and specialized T cell. During future exposure to the coronavirus, these antibodies and T cell will attack the virus, preventing the development of COVID-19. And finally, thank you.